In the next activity, we're asked to do the same thing, just looking at it in a little bit more detail. So you can either start with pictures if you're more comfortable or more visually oriented, or you can start with the words. I tend to do better with pictures, so I was just going to start there. And basically, I look for where DNA is not in little bundles or they don't look like little X's. So that's interphase. In prophase, the nucleus is starting to disappear. In metaphase, they're lined up in the middle. In anaphase, they're moving away from each other. In telophase, notice that it's starting to pinch in, but not very much because cytokinesis actually happens at the same time. But the chromosomes are at the two ends. And then right before the cell splits in two, you can see it's almost splitting. That's cytokinesis, and that's division of the cytoplasm. If you're looking at what goes on in each phase, interphase used to be called the resting phase, and it's not resting, but it's because when scientists looked under the microscope, that's what it looked like they were doing. When you look at prophase, you're going to see that chromosomes are condensing, like we just said. You're looking for chromosomes lining up in the middle in metaphase, and they're moving away from each other or being pulled apart in anaphase. The chromosomes are at the two ends in telophase. And then finally, like I said before, you see them pinching in or forming a cell plate if it's a plant cell in cytokinesis. If you'll pause and correct your answers before you move on to the next activity. In the next activity, you were asked to do the onion root tip lab, and so it took you to an online website since we couldn't use the microscopes at home. And on this website, basically, you were looking at cells that had been given a chemical and they were frozen or stopped in certain phases. So this is what it would look like if we looked at all of them at once. And so when you got to the activity, after reading some background information on it and about the phases, it asked you to fill in the table, and you didn't have to make it because I already had it made for you. And so you're just going to identify which one of these phases each of the cells is in. So for each picture, it says click on the phase. So this one, they're lined up in the middle. So I clicked metaphase. In this one, I can see that I don't see the little bundles, so I think it's interphase. And it asked you to do this for every single one. And it told you in the table, or if you looked here, there were 36 of them. So you kept doing this until you got to the very end. And you hopefully noticed that there were a lot more cells in interphase than in the rest. So as you went through and counted, hopefully you noticed that when you got to the end, you went back and checked that you did get all 36. And these were the numbers that you got: 20, 10, 3, 2, and 1. And then if you followed this problem down here to find the percentage, we take 20 divided by the total number of cells, 36, multiply it by 100. So 56% of the cells were in this phase. And then notice we get fewer and fewer as we go down. And that just has to do with how the cell cycle runs. And that's back to the activity I actually skipped by accident. So we'll go back to that in a minute. And then again, you're going to put these in the correct order. And notice that interphase, you cannot see the little distinct bundles, but you can in prophase. Metaphase, they're in the middle. Anaphase to two ends. And telophase, they're split. Anaphase, they're moving apart. And then telophase, they're at the two ends. Again, because I did accidentally skip it, if you will look up here and pause this and check your answers to the previous activity, and then move to the second slide and check your answers to the second slide as well. This basically is just giving you a way of looking at the breakdown of the cell cycle, whether it is in a cycle, which is what actually happens, or if it's easier for you to break it down and think